part of the corn board, but you all probably provide into that. So we thank the corn board for sponsoring. They sponsored every year that we've done this, so we really appreciate their support. And yeah, I think with that, I'm going to go ahead and kind of introduce kind of what we're doing today. So it's all on crop nutrients and diseases because, you know, 2 hours you can learn everything about crop nutrients and diseases. So that's the plan for today, right? See, it only took Kyle an hour to learn all of the diseases there are. So, but no, we hope that this can provide you a little more knowledge on maybe what you're already coming in with as far as all of the gamut of issues that can be out there in that cornfield. And so the first up is Kyle Broderick. He's a coordinator of the Plant and Pest Diagnostic Clinic. And then Ellen Paparosi will be later on talking more about nutrients. And I also want to introduce Melissa Bartels. Or is it Bartels? I'm not sure how you say it. Bartels, that's what I thought. She's a new educator located in Butler County, but she's from Waverly area, I think. And so she does a lot with plant physiology and some of those things. So she would also be a local contact in the counties to help with some of those issues. So I was glad she was able to come today. So maybe at break you can visit, especially if you're located in that end of the county, be kind of close to her. So with that, I'm going to go ahead and turn over Kyle. You can probably do a better introduction of yourself than I did, but I appreciate you coming in. And again, feel free to jump in and ask questions as they come up.
Yeah, exactly. All right. Thank you, Tyler.
Give me the clicker here if you need it.
All right. Well, yeah. So thanks for showing up this morning. Really appreciate all you guys being here. Just raise of hands. Kind of curious what you guys grow. So corn, most everyone. Beans, most everyone. What about wheat? Got a handful. All right. All right. Oh. So really, regardless of whether it's disease management or nutrient management, you want to know what you're dealing with before you try to fix it. Because you go out and you look in your field and there are a lot of things that can make those plants look yellow or maybe a little bit purple or just off. And identification is by far the most important step to any sort of management. If you're not putting the right treatment out there, it's not going to help, not going to be effective at all. Now you're just wasting money. But there are two types of disease that, at least as a pathologist, we deal with. We say that there are two types of diseases. Ellen may disagree with some of this, but so we talk about abiotic diseases and biotic diseases. And so the abiotic diseases, that's going to be some of the nutrient issues, whether it's drought, heat, things, a lot of those environmental problems, we consider those to be abiotic or non-living diseases. Conversely, we also have our biotic diseases. And so those are going to be diseases caused by living agents. So that's your fungi, bacteria, viruses, nematodes, etc. And really quick terminology. Just a lot of people will use these two terms interchangeably, but there are some slight differences. And so a sign versus a symptom. And so a sign is the actual visible evidence of that pathogen. And so if you have a fungal pathogen and you're seeing some of that cottony mycelia growing up out of the leaf, that's a sign. Or if you have a tree conch, that fungus, that fungal shelf, that again is going to be a sign. Otherwise, we have our symptoms, and the symptom is just the plant's response to the disease, or the plant's response to the infection. And a lot of times those symptoms then will just be yellowing, or maybe you'll have a little bit of a lesion with a halo, something like that. And I just wanted to draw your attention to the size of a lot of the things that I work with in the diagnostic clinic. Here we have our prototypical plant cell. It's more or less a box, but starting from the biggest, we have our nematodes, and this is just the nematode head is typically the only thing that will enter that plant cell. And then moving down, we have our fungal hyphae or fungal mycelia. And both the nematodes and our fungal mycelia, those are both pathogens that will actually break through that cell wall and that cell membrane in order to absorb the nutrients. As we move down, we have our bacteria. And so again, our bacterial cells, much smaller than even the plant cell's nucleus, which we have right here. And even smaller than our bacteria are our viruses and mycoplasmas or phytoplasmas. And in comparison, for ideas of some of the sizes of our viruses, they're pretty small compared to those plant cells. And so I was talking about how the nematodes and fungi, they'll kind of break in and take their nutrients. And so I like to break things down into groups as best I can. And so I always think it's a great way to think about pathogens as you have your thugs and you have your cons. And so your thugs, those are going to be those pathogens that they'll just bust right through the cell wall. They do not care about setting off any alarms. They're going to get in, steal the nutrients, and before the plant has time to respond, that fungus or that nematode has already gotten what it wants and it's moved on. Otherwise, we have our cons and the con artists. And so our bacteria, our viruses, we tend to think of them as kind of con artists, where they're trying to trick the plant, and so the plant doesn't necessarily know that they're in there at first. And they can be taking as many nutrients as they want, but by the time the plant realizes that there's a disease there, by that point it's too late. So, starting with our biggest group, are nematodes. Most nematodes will feed on the below ground parts of roots, but we do have some that feed on the foliar parts as well. Oh, if any of you guys have pulled up any soybean roots and seen some of these white or kind of cream-colored little oval sacs on there, that is soybean cyst nematode. And in case any of you guys have bean fields and have never tested for soybean cyst nematode, you're already paying for a free test with your soybean checkoff dollars. And so afterwards, come talk to me and I'll find a way to get you guys some bags so you can test for those. But the cyst nematodes are just one type. A lot more of our nematodes are kind of like this, where they are more vermiform or worm-like. And what they'll do is they'll just stick their head, and then here we can see the stylet that they have right here, and that stylet is used to actually penetrate through that thicker cell wall to absorb the nutrients. Getting a little bit smaller, we have our fungi. So our fungi are filamentous organisms that lack chlorophyll. They are kind of similar to plants, though, otherwise. And most fungi reproduce via spores. And so when I get a sample into the clinic, I'm actually looking for the spores that they produce. And here we have a video or a picture of some of the different spore types that we have. But those spores are what I'm actually looking at. Unfortunately, I can hold up an ear of corn like this, and most of you guys probably have some idea what this is. It looks a lot like fusarium. Fusarium on corn, pretty typical. You get that kind of matted up towards the top. Unfortunately, though, the pathogens don't read the textbooks. And so they don't really know how to infect. So symptomology is not the end all when it comes time to diagnoses. And getting even smaller, we have our bacterial pathogens. So bacterial leaf streak, that's one of the big bacterial diseases that has recently shown up. And one of the great things about bacteria, it's really cool to look at, is you can cut it, and when you cut one of those lesions and put it underneath the microscope, you can see all of those bacterial cells just streaming out. And one of the great things about bacterial leaf streak, this new corn disease, is it gushes. And so there are some pathogens where you cut it, you look for this bacterial streaming, and it's kind of weak. You can't tell too well. Luckily, BLS, it is a gusher. And so right here, we have tens of thousands, if not millions, of bacterial cells that are streaming out of those, of the plant tissue. And we do have a few different shapes of bacteria, but typically we will plate the bacteria onto media, and they'll form colonies kind of like this, kind of mucoid type colonies. As we get smaller then, we have our viruses and our phytoplasmas. And so viruses, depending on the argument, may or may not be technically alive. Again, that goes into a metaphysical discussion of philosophy and what is life. But Viruses actually utilize the plant's own metabolism and reproductive system, or just the plant cell, in order to complete its life cycle. And so viruses can't complete their life cycle without having that plant host. And another one is our phytoplasmas. And phytoplasmas are one of the newer groups of organisms that we have. For a while, they were classified as mycoplasmas, but really they're pathogens that are kind of a cross between a fungus and a virus, but not quite. So really can't do any sort of pathology talk without bringing up the disease triangle. And anyone who's heard me talk, I apologize because this is repeat, but you really need to have these three components all coming together at the same time in order to cause disease. And so we have our conducive environment, a susceptible host, and a virulent pathogen. And without all three of those, we won't have disease. But now there is a fourth component that is almost more important, and that's timing. And so it's not just enough that we have these three portions or these three components, but they all have to be occurring at the same time. And the longer that they're occurring at the same time, the greater impact that this disease will have. And I just want to draw your attention to the updated plant hardiness zones, just to kind of show what things are doing in this country. A lot of diseases are made worse by any sort of plant stress. And one of the things that we're going to see more and more of probably are some environmental stresses, primarily due to our plant hardiness zones really moving north. And so it's a lot warmer here. The moisture patterns will change. A lot of those environmental conditions will impact the diseases that we get. So when we're looking at diseases, or if you've ever sent a sample into the clinic, one of the first questions that I always ask is, well, what's it doing in the entire field? So we always like to start big and then narrow, whether we're looking at entire symptom distribution or even on a microscope. We always start with the lowest magnification and then kind of hone in from there. But I always like to figure out what the disease is doing on three different scales. And so first we have our fields, the field distribution, and then the plant distribution, and finally, where on the individual leaves are we seeing these symptoms occur? And a few things to be thinking about when looking at symptom distribution. Are the symptoms random or are they in a pattern? Disease tends to be fairly random. If you have a pattern that you can correlate to, say, irrigation tracts or something like that, might not be disease. Maybe there's some other stress that's causing an issue there. Also, are there the areas in the field that are worse? Is there some sort of issue with soil moisture there? Are they in lower areas or are they in higher spots? And finally, what's the relationship to other features in the landscape? Do you have some bins in one corner of the field? Or do you have a fence line? Things like that. All of these things can impact the microclimate, which then impacts the individual diseases that will occur there. So looking at field distribution patterns, one thing is, or a few things that are really important to remember, and so really it's just keeping a good field history. And so knowing kind of when you planted and what you planted each year, if you've done any sort of soil modification, sometimes we won't see results from some soil modifications for 2 or 3 years later. And so it is nice to know what's been done in the field. Also, what's the disease history in the field? Do you have that one corner that seems to always get Phytophthora? That's important to know. And also, the chemicals that have been applied for the past couple of years are extremely important as well. One of the issues that we saw last year is we had some fall applied herbicides that didn't break down the way they were supposed to over the winter. And a lot of that was due to the weather, just last year's winter that we had. We didn't really have the moisture that was expected. A lot of those herbicides weren't able to break down. They're a little bit lower in the soil profile, but then and I saw this issue in beans quite a bit more, but really once those roots started to grow down, they came in contact with the herbicide, And then we started to see some phytotoxicity occur. The other thing is, are there any obstacles in your field? Do you have a tree line? That's really going to influence the moisture. Anything that influences moisture is really going to influence the diseases that occur. And so as we have our kind of even or more uniform patterns, we also like to think, okay, where are they occurring? Is it something that's just kind of at the entry, the field entranceway? Is that where most of the issue is? So where a lot more equipment's traveling through? Or are most of the symptoms based on one side of the field? And if they are on one edge of the field, how do they look? Do they end? Is there pretty much a straight line where you see the symptoms stop and then you have healthy plants? Or is there more of a gradient, kind of like we have right here, where it's worse, but then it gets a little bit better the further out into the field we move? All of these things are very important to help identify diseases. Previous uses of those fields too, though. A couple years ago, maybe you split that quarter and did half corn, half beans, and then next year you decide to do the whole quarter into corn. Well, that's going to change. That's really going to change some of the soil, and so you can easily have differences that are primarily based on the previous year's crop. So really, all of these things go into knowing your site history. And I ask a lot of questions about site history. And so including all the chemicals, planting things, the different cropping systems that you've used. I also really like to know what the soil pH is, what the soil type is, because all of these things really play a role when trying to figure out what exactly is causing the plant to look sick. Otherwise, we may have some very uniform patterns in the field. If you ever see something like this, where it's just one row that's affected, most likely that's going to be some sort of planting issue, something like that. Again, disease tends to be random. Disease really doesn't follow straight lines generally. It can, but often any disease that follows a straight line is going to be kind of a weak disease, a secondary pathogen that is utilizing that stressed plant, and now it's able to cause disease. Additionally, any sort of field operations that you've done. If maybe you did some tillage just around the border, that's where you're seeing the issues. Again, all of these field distributions are very important. But this is what I tend to think of as far as a disease distribution, where we have these kind of irregular patterns in the field, and there will be kind of a gradient there in the field as well, where there will be one kind of the center, that's the hot spot, and moving out from there is where we'll see the diseases occur. However, if you have any sort of topography changes in your field, those often turn into kind of irregularly shaped patches where you're seeing those symptoms occur. And so that's another thing that's really important to let me know. But as we zoom down, now we're looking at symptom distribution on the plant. And so where on the plant are we seeing issues? Is it in the root system? Is it in the lower canopy, all the way throughout the canopy? Or are we looking at the reproductive parts of it? Most diseases, or most pathogens, they have a certain part of the plant that they prefer to infect. And so knowing where on the plant we're seeing these symptoms occur, that can really help me narrow down what's causing the issue. And one of the biggest things when looking at distribution within the plant, first, know what the healthy plant is supposed to look like. And I can't stress this enough. With all the different varieties and hybrids that are out there, sometimes there will be maybe some color changes, and that's a known variant that this hybrid will do. And that's not necessarily a disease, but that's to be expected. And so it's really important to know what the healthy plant is supposed to look like. But I also want to draw your attention to that bottom bullet point. And a lot of times when we're seeing foliar symptoms, that's not necessarily a foliar disease. That disease is probably a lot lower down. Maybe it's in the root system, or in the case of corn, We have some sort of stalk rot that has caused basically this entire bottom part of the crown to rot away, and maybe we have a few different fungal pathogens inside of there. That also causes a lot of foliar symptoms too. And it all goes back to, are the symptoms uniform or are they kind of random? If you have the same size spots on every leaf, that's probably not going to be a disease. That would mean that every spore had to land at the same time and grow at the same speed. So generally, if you see a situation like this, now we're going to be thinking maybe this is a herbicide issue, where we have the water droplets that have landed there, and that's what's causing the bleaching. Something like this, where we have the different lesions that are all different sizes, different parts of the leaf as well, that is much more common with disease. We may also see something like this, where we have some interveinal chlorosis, or the issues where they follow the vein. Certain pathogens, they will travel along the veins, whereas other pathogens can't cross those leaf veins. And when dealing with a nutrient deficiency, there are really two important questions to ask. And so first, is it the top or the bottom of the plant that's affected? And then the next question is the dominant symptom chlorosis, or is there something else that we're seeing? Maybe there's some red along the base. Maybe that's more dominant than the chlorosis. And again, you guys should have these handouts, but just a quick map or diagram of a plant and kind of where we see some of these nutrient deficiencies occur. And again, we have a few more here. But one of the issues that we saw a lot with the plant diseases last year, we saw a lot of seedling rots, or a lot of issues with early season issues. And that was all because of you think about the last spring that we had. We had an extremely cold and wet April, and then May came, and we had 100+ degree weather at the end of May. And so a lot of plants, as they were just kind of getting the start out of the ground, they were experiencing cold stress, flood stress, and heat stress once they got out of the ground. And all those different conditions led to a lot of different seedling diseases last year. And these are our main 4 soil-borne diseases, or the main root rots that we see in an agronomic setting. Pythium, Phytophthora, Rhizoctonia, and Fusarium. Luckily, corn does not get Phytophthora, so if you only grow corn, you can kind of knock that out. But anyone who does beans, I'm sure that you've dealt with Phytophthora in your life. But Pythium both Pythium and Phytophthora, they're kind of our water molds. And so they prefer that soil to be quite flooded, because they'll actually need to swim through that soil profile in order to reach the plants. We also have our Fusarium. Fusarium really can infect, if it's wet, it can infect. There are certain species of Fusarium that prefer drought conditions to cause infection. Fusarium is really one of those wide-ranging pathogens that can cause disease over really a broad range of conditions. And the last one on here is Rhizoctonia. And Rhizoctonia tends to like the soil damp, and so not flooded, but also not bone dry, and kind of warmer temperatures. But if you look, so really every sort of soil and environmental condition that you would expect to see early in the growing season, we have a soil-borne pathogen that can cause disease in those conditions. And they can be pretty difficult to figure out. And early in the season, you really want to make the decision, do I need to replant as early as possible? And so we always recommend kind of just looking for seedling skips. And this picture is a little bit difficult to see, but we have our rows here. And here we have the few corn plants that have come up, but we also have these big skips in there. And when you're seeing something like that, it's a sign that, okay, maybe we are dealing with some sort of soil-borne seedling disease. And when looking at the seedling diseases, I didn't throw Phytophthora up here because that tends to behave a little bit differently. But there are some features or the way that these diseases cause infection that can help you tell them apart. And so Pythium really tends to girdle the root from about the soil line down. And if you have an infection with Pythium, if you can just kind of run your fingers along that root system, and you can probably pull off the epidermal layer of those roots. They really just slough off with the Pythium infection. Rhizoctonia tends to cause lesions right about the soil line, a little bit higher up on the plant stem, and it'll look healthy probably down below and up above. Tends to be Rhizoctonia. Fusarium is a taproot fungus, and so anytime you have an issue with Fusarium, that one's going to be, you'll probably have a healthy root system higher up, but as we go down into that taproot, that's where we're just going to start seeing a lot of the lesions occur. Anyone who's dealt with sudden death of soybeans and has pulled up any of those roots and there's just no taproot whatsoever, well, sudden death of soybeans is a Fusarium disease, and it just completely rots off that taproot. As far as managing these seedling diseases, get a lot of questions about, do I need to use a seed treatment fungicide? And the answer to that is maybe. But again, this all goes back to your site history as well as your agronomic practices. If you're dropping a lower number of seed, well, maybe now you're going to want to use one of those seed treatments so that you're protecting every seed that comes up out of the ground. If you're going to be planting a little bit heavier population, maybe you don't need that seed treatment because it's okay if you lose a couple of plants because you have enough others out there. Again, a lot of these management recommendations go back to your individual agronomic practices. Moving on to some of the diseases that you guys have seen or will see this next year. Get a lot of questions about bacterial leaf streak and gray leaf spot. And I like to think that I can tell them apart pretty well, but there were 7 times last year where I got a sample and I had a colleague and we'd bet $1 on it. And so, okay, it's very early lesions of either BLS or gray leaf spot. Which is it? We put $1 down each time. And I lost $7 last year because I was mistaken. Because it was, again, the lesions looked exactly like BLS, turned out to be gray leaf spot. They don't read the textbooks. But a few ways that you can tell these very similar pathogens apart. With bacterial leaf streak, if you look at the margins, we tend to get these kind of wavier margins that we get. With gray leaf spot, it's much more rectangular. Again, the early lesions, very difficult to tell apart, but once they mature, then you kind of can. The other thing that I always do is I always backlight the samples. And so I like to hold them up to the light and see, okay, what does it look like with the light shining through? And BLS, or bacterial leaf streak, has very striking, it's almost neon colors when you hold it up to the light. Compare that to gray leaf spot, where there's some brightness around the margin, but it's just not as colorful as bacterial leaf streak. But the big one with these are the wavier margins. Why is it important to be able to differentiate these two diseases? Well, one of them is a fungus, the other is a bacteria. So one of them is easily controlled with any of your standard fungicides. Bacterial leaf streak, we don't really have any good products that the university has shown that can help manage that disease yet. A couple other diseases that look very similar and I get a lot of questions about will be northern corn leaf blight and Goss's wilt. Last year was a bad year for both of these diseases. We saw a lot of northern corn leaf blight in the clinic and actually saw a fair amount of Goss's wilt as well. But Goss's wilt, that's the bacterial pathogen, and that one tends to be, whereas northern corn leaf blight has these larger lesions, they're pretty cigar-shaped, tend to have a kind of a dark margin with them. But once we leave that lesion, the plant or the leaf looks healthy almost immediately. with a lot of our bacterial diseases, they'll do what we call water soaking. And so where it's the lesion, we have the lesion right here, but then as we move out past the lesion, the leaf still doesn't look terribly healthy right away. It maybe just looks like it was soaking in water, something like that. And that's the high number of bacterial pathogens that are causing the disease. One of the other diagnostic features of Goss's wilt are the freckles that we tend to get inside of those lesions, or those black specks. A lot of times people, if they see those black specks inside of a larger lesion like that, they'll say, oh, all right, we have Goss's wilt. Unfortunately, if you have a lesion from northern corn leaf blight, well, you now have dead plant tissue in the middle of that lesion. And there's a lot of different fungi that can colonize dead plant tissue. And so it's not uncommon to get northern corn leaf blight lesions that, and here you can kind of see it right here, but there's a lot of dark specks in there. And that's all been colonized by secondary fungi. And so without looking at it underneath the microscope, really difficult to tell those apart. And again, the reason that it's important to tell these two apart is their management is completely different. For Goss's wilt, you really need to be looking at some of our larger agronomic practices, whereas northern corn leaf blight responds very well to a lot of our fungicides. Next, the rusts. So common rust versus southern rust. You guys, anybody want to take a guess as to which is common, which is southern on here? No? Yeah? All right. I see a hand. Oh. Shoot. Well, I failed. But yeah, so we had southern rust on the your left, and then common rust on the right. But those two diseases can look very similar out in the field. As far as management for common rust, a lot of that's going to be just using a resistant variety. We tend not to see as many issues with common rust where fungicides are warranted. Now, southern rust, yeah, do you have a question? Well, yeah. On southern rust, you know what you're talking about. What do you call it? The zone that's moving north, obviously. The hardiness zones. Yep.
You think southern rust will move farther north than that?
Yes. Yeah. So yeah. Yes, I do. So the question was about, as our plant hardiness zones are all moving north, we're having warmer temps a little bit north, higher into the continent. Will we see more southern rust a little bit in some of the northern climates as well? And I would expect the answer is yes. Southern rust, it likes but you know, kind of mid 80s to low 90s, kind of later in the season are the temperatures that it prefers. And so the hot, and especially at night, those nighttime temperatures are extremely important for a lot of our pathogens. But the farther north we get, the more common it is for it to be 85 to 90 degrees at night. We're going to see a lot more issues with southern rust, I would expect. Now, last year, we had southern rust show up the earliest that we've ever seen it in the diagnostic clinic. In about 25 years of keeping records, last year was the earliest that it had shown up. We were nervous, but a lot of that is because you think about the weather that we had last year, and we had 4th of July weather first part of June. And so we had those high temps, those high nighttime temps, and plenty of moisture early in the season. And so the rust, and rust is one of those diseases that blows up from the south. And so as people down in Texas, Oklahoma, Kansas were dealing with it, it was just slowly making its way up. And so we were really nervous about southern rust last year. Dr. Jackson, Zims, and I, we were expecting to see just tons of it. But then as we moved into the growing season, kind of in late July into August, temperatures dropped a little bit. It got quite a bit cooler. Really wasn't conducive to southern rust. And so I don't know if any of you guys had southern rust outbreaks in your fields, but we really kind of escaped it last year for the most part. And it was all because of that late season weather that we had. And so as far as telling the two apart, southern rust tends to have more orange or tan pustules, where common rust, we tend to think of them as kind of brick red. The other thing is, where are we seeing those pustules? And the pustule that I'm talking about is just one of those little spots there. All right, test time. Are these spots a symptom or a sign? Sign. Yep, I heard it. Yep, because that's the visual manifestation of the pathogen. But those pustules with southern rust, we tend to only see them on the top of the plant. Whereas with common rust, we tend to get those pustules both on the top and on the bottom. But remember, they don't read the textbooks. And we had a couple samples that came into the clinic last year, and it was southern rust as soon as we looked under the microscope, but we had pustules on both the top and the bottom of the leaves. And with looking at common rust, the optimal temperature is a little bit lower as well. That's why we tend to see common rust showing up a little bit earlier than southern rust in many situations too. Have a picture of a leaf right here. And we had a lot of samples that came in looking just like this early in the summer last year. And for the life of me, we could not figure out what was wrong. We would try to isolate fungi out of there, couldn't get any fungi. We'd go for try bacterial diseases, we couldn't isolate any bacteria. After talking with a few other agronomists, what we realized is it was a highly resistant hybrid to common rust. And what we're seeing here is just a hypersensitive response to that common rust fungus. And so the fungus landed on the leaf, tries to cause infection. What does the leaf do to prevent infection? It just kills all those cells right in the surrounding area. Once those cells are dead, now that pathogen can't move, it has no food source, it can't reproduce. Some of our, moving into some of the soybean diseases then that we saw a lot of last year is frogeye leaf spot. And I don't know if any of you guys dealt much with frogeye. It's more of an issue kind of in the northern part of Nebraska, but we are seeing it really across the entire eastern third of the state and kind of just slowly moving west. It's a fungal disease, tends to be much more severe in our no-till or continuous soybean fields. It is a residue-borne fungus, so it just survives in that dead plant material. There are some genetic resistance available, but typically frogeye responds very well to fungicides. And so especially our strobilurin fungicides or our QOI, the quinine outside inhibitor fungicides, work quite well for frogeye leaf spot. Unfortunately, sorry, I got ahead of myself. But here we have, you can just see, the evidence. Whereas non-treated leaves, lots of lesions. This was a low rate of the strobilurin fungicide as well. But unfortunately, as with a lot of our fungicides or a lot of our fungal diseases, we've been spraying a lot and maybe not spraying things in the smartest way. We're dealing with the same issue with a lot of our weeds. But we are having strobilurin resistant isolates of the frogeye fungus show up. And that's fairly concerning because especially down south in the Missouri Delta region, frogeye is a big issue. And as we expect things to get a little bit warmer up through Nebraska, we're now going to be having weather conditions that are much more favorable for frogeye as well. So they're already dealing with resistance way down here. And unfortunately, this map was released in late 2018. And in about November of 2018, it was reported that there were 4 counties in South Dakota that also tested positive for the strobilurin-resistant frogeye. Yeah, question.
Are you concerned about triazole resistance developing? Because we're putting one mode of action on this now.
Yeah. So I had a question about concern about triazole resistance occurring in frogeye. And the answer is not as much, at least not as much as the strobilurin resistance. And so to develop resistance to a strobilurin fungicide, most strobilurin fungicides have a single point that they attack. And so it just takes one mutation in that fungal genome for it to now become resistant. Whereas triazoles, they tend to affect kind of a broader part of that plant cell. And there's a few more mutations that have to occur in that cell for it to become resistant to triazoles. But so yeah, triazoles, they are kind of on the lower end of the resistance. But really, the big thing that I would like to recommend is just using multiple modes of action as best you can. And so, all right, maybe the strobilurins didn't take care of it, but they took care of a lot of them. And now we have the triazole that's able to come in and take care of the rest. But those combination products, very effective when dealing with any sort of resistance issues. So, had mentioned Phytophthora, root and stem rot earlier. This part of the state last year, we had a lot of moisture early in the season, a lot of flooding issues kind of in through June and July. Really saw Phytophthora take off, in a couple instances where we had field-wide failure of that Phytophthora resistance. So Phytophthora is one of those soil-borne diseases that we tend to recommend that you manage with the use of resistant varieties. Seed treatments are somewhat effective, but anytime you have a seed treatment, that's only going to be active in the soil for about 30 days. And at the end of that 30 days, now you still have that fungus there in the soil, and it's going to be able to infect. Whereas Rhizoctonia, Pythium, those can infect larger, more mature root systems very well. Phytophthora can. And so, one of the issues that we saw last year was, where we had entire fields that were failing with their resistance to Phytophthora, unfortunately. But when looking in the field, it's pretty common that you'll have a line of healthy plants and then maybe 3 or 4 just right in a row that are all shriveled, dead, and brown. But one of the features that I like to look at is typically, and I don't have a good picture of it, unfortunately, but there will be some black discoloration that kind of starts from the soil line up, and typically that black discoloration is only on one side for Phytophthora. The other thing that Phytophthora does is it causes this mass wilting of the plants, but the leaves and petioles remain attached. And so if you have this wilting, but the leaves have dropped off, well, now it's probably not going to be, or at least Phytophthora is not the only issue that you're seeing if the leaves are dropping off. And had samples that came in that looked a lot like this. So we have wilty plants, leaves are attached, the petioles are attached. We have a line of healthy plants, then we have a patch of dead, dying plants, then healthy plants afterwards. That is textbook Phytophthora. Unfortunately, we couldn't get Phytophthora out of any of these samples. Start cutting them open, and we'd start seeing that gall midge. So if any of you guys were here for Justin's talk a week or two ago, he probably talked a little bit about this. Just show of hands, how many of you guys think you dealt with the gall midge last year? Quite a few. And unfortunately, there's so much that we don't know about this pest, but it causes symptoms that are almost identical to a few different fungal diseases. In addition to Phytophthora, when we have something that looks like this, where there's going to be just a big canker or a big lesion a couple inches up the stem line, now that's almost identical to stem canker. And so that's another fungal disease. Looks very similar to the gall midge, but it's not. So again, before you ever start managing for something, it's critical to know what your main problem is. And starting in about late August, early September, started to get phone calls. Started out 1 or 2 phone calls a day. By the end of the season, it was probably up to 5 to 7 phone calls a day. My soybeans are moldy. The stems are completely black. yielded about 10 to 15 bushels less than was expected. And unfortunately, that was just kind of last year was a bad year for beans. Some fields did great. We saw a lot of issues. We saw a lot of fields that almost had record yields. But we saw a lot more that came in looking like this, where yeah, the stems are completely black. We have a whole bunch of lesions there on the pods. Crack those pods open, and we look at the seeds, and the seeds are shriveled, moldy, have spots on them. Just really low quality seed. And while the discoloration and the shriveled seed, it is all a biotic disease that's all caused by fungal pathogens, most of the reason that we saw any of those issues was because of the late season weather that we had last year. So think about what happened kind of middle of August and through September. The sun kind of went away, it got really cloudy, and it got wet and cool. And so as though we even had some instances of beans sprouting in the field where we had the pods would crack, moisture would get into those pods, and then we'd start to see a radicle come out of those seeds they were trying to germinate in the field. Had never seen that on soybeans in my life until last year. But I had mentioned it's all because of the weather that we had at the end of last season. So if we look at this precipitation map of the last half of last year's growing season, especially in our part of Nebraska, right here, we're looking at about 130 to 200% of our average moisture at that time. Also, we had it was fairly cloudy during that time too. And so any moisture that dropped, it really wasn't evaporating, wasn't being utilized by the plants, it just kind of stuck around. As though the beans inside of the pods then it was a perfect conditions for them. The pods would crack, fungus gets in, moisture gets in, but you can't get into the field to harvest. And so you had to delay harvest a little bit. And meanwhile, those fungal pathogens just keep causing more and more disease issues. This is a problem that is, while it's new to Nebraska, down in the South, this is something that they deal with every third or fourth year. They expect to have just major issues with moldy soybeans. And when you think about the climate down south, it's going to be much more humid, it's going to be warmer, but that excess moisture later in their growing season makes it that much easier to cause these fungal issues. Had a lot of people last year, the people that did have a great soybean yields, found out that they were some of the ones who ended up doing a late kind of an R5 fungicide application. Really paid off last year. Will it pay off next year? You know, unless we have weather like we did last year, it's really hard to say. And so if you do have a history of some of these diseases in your field, if you have a lot of frogeye showing up, okay, maybe go ahead and do one of those later fungicide applications. But as we get later into next season, and you hear your neighbor telling you about how their R5 application just caused their soybean here, whereas frogeye leaf spot doesn't do that. Frogeye will cause some spots on the leaves, but we don't get that purpling that occurs. But we had quite a few samples last year that came in, and I was able to isolate that frogeye pathogen from the seed. Not a major issue though. And the, I'm sorry, what was the next, the second part? Oh, okay. Yeah, the short season bean. And the answer is, yeah. Well, if the shorter season bean that you have, you may end up escaping some of these late season diseases that we saw so much of last year. Not always, because weather plays such an important role in that, but the earlier you're able to get those beans out of the ground last year, we did escape a lot of the issues that we saw. So what was causing the stems to be black, moldy? Well, the big one that we saw a lot of was pod and stem blight. And so earlier I mentioned stem canker, and next I'm going to talk about phomopsis. Phomopsis, stem canker, pod and stem blight, those are all names of the same thing. And so those are all caused by Diportha species. So fungi that are in the Diportha genus. But pod and stem blight, one of their great diagnostic features of it is they have these pycnidia, and the pycnidia are just kind of our black, they're our fungal fruiting structures, almost look like black pimples. But the pycnidia will show up really in a straight line. And typically it's going to be right beneath the node is where you tend to see those lines of pycnidia occur. This one, we tend not to think of it as causing major yield losses, anything like that. It's just one of those late season diseases that last year we had the perfect storm for, and it was really able to take off. But we did see issues with phomopsis. And so phomopsis seed decay, this one is one where you'll not only have yield issues, but you take seed that looks like this into the to the elevator, and you may end up getting docked for it. We did have a few instances of people getting docked for poor quality seed last year. Unfortunately, a lot of it was due to phomopsis. And then Cercospora leaf blight and purple seed stain. Both of those are caused by the same pathogen, Cercospora kikuchii. But the important thing about Cercospora leaf blight and purple seed stain is while there are resistant varieties available for this pathogen, the resistance for the leaf blight is not the same resistance as for the purple seed stain. And so generally, the leaf blight tends to be not cause as many issues. Almost just looks like sunburn, kind of have some bronzing on the top of the leaves. But when you start having a lot of seeds that have this purple discoloration on them, now that can really cause some issues once it comes time to market those beans. But we do, one of the things about purple seed stain, if you do deal with it year in and year out, maybe be thinking about one of those R5 fungicide applications. We have seen pretty good control of purple seed stain with those late season fungicide applications. Unfortunately, it's a Cercospora fungus. Earlier I was talking about our other Cercospora, Frogeye leaf spot, and we already have our QOI resistance documented. While we haven't found it yet in this Cercospora fungus, it is possible that we will start having resistance issues show up. So again, just be smart about what you're spraying. Anthracnose stem blight then, saw a lot of that, and this one tends to cause just larger black splotches up and down the stem. Again, we really don't think of anthracnose as causing major yield losses. If it comes in early in the season, it can, but a lot of the diseases that I got so many calls on at the end of last season are things that generally shouldn't cause many issues. Last year, everything hit perfectly, and they did though. As far as any kind of switching gears to the wheat, we did have a few wheat growers. For those of you who grow wheat, did you guys deal with black chaff or black bacterial streak last year? Or not that you saw at least. We saw a lot of bacterial diseases last year, and in wheat, tons and tons of black chaff or bacterial streak. And so here we have kind of some earlier symptoms. You can see, notice how that leaf is kind of shiny. That shininess is that bacterial exudate kind of coming out of that leaf. That's making it shiny. And here we have our much later symptoms as well. But early on, just kind of those nondescript yellow to brown lesions can look like a lot of our wheat diseases. Tan spot can look fairly similar to this. But cut it open and we see just tons and tons of bacteria streaming out. So last year was a bad year for black chaff. Unfortunately, there's not a lot that can be done. When the environment's perfect for it, we're going to have a lot. We're just going to have a lot of it. This pathogen can be seed-borne, and so we recommend that if that you try to purchase your seed try to purchase disease-free seed, but that can be really difficult when dealing with this pathogen. So early in the season, when it's on the leaves, we call it bacterial streak. The black chaff phase tends to be more in the heads, and this is when people start to get really concerned then. When the heads just turn black, and then you look up those awns, and it's hard to get a good picture of it, but we can kind of see it in this one. But then on those awns, there's just going to be a black line across those awns. And that is one of our diagnostic features for black chaff. And just a little bit about the clinic. If you would like to use us this upcoming season, kind of the things that we do, I provide identification and management recommendations for really both abiotic and biotic diseases. Any insects or arthropods, we can identify those as well. And so just feel free to send in any samples. Any unknown weeds that you have, we can figure out what that is, and so you know what herbicide program you need to deploy. As far as herbicide injury, got a lot of calls on that last year, and we only diagnose herbicide injury on a visual basis. We're not set up to do any sort of residue analysis, and so we can only look and say, oh, well, based on the spray program provided and the symptoms, yes, this could be caused by the herbicide, or no, it's unlikely to be caused by the herbicide. As far as what's not provided, again, the residue analysis for herbicide, but we're also not able to do any sort of soil testing, and we're just not set up for that, unfortunately. Nor are we set up to test for mycotoxins, and so that's going to be a third-party lab that would need to be contacted. If you'd like to do any mycotoxin tests, you can call me, and I can send you to another lab, or kind of give you a few to check out. But we're not able to do that in Lincoln. And when you send a sample in, we really want to make sure that you follow just a few basic rules. And this is one of the best samples that I got in last year. And so we have the whole plant, we have the root ball as well. And so they just dug that plant up because, again, a lot of foliar issues. It's the root that's mainly where the issue is started. But so send in the whole plant. They sent in multiple plants. And so if you have 140,000 beans per acre, please don't send me one soybean. You can probably afford to send in 3 or 4. But so I have multiple plants. There are some healthy plants in there as well, so I can compare, all right, the symptoms that I'm seeing in the clinic, how do they look to the normal plants? And that root ball is then contained inside a plastic bag to kind of keep a lot of the soil contained. As far as what not to do, really don't add water. A lot of people will do that. When a sample comes in that looks like this, I can't do anything with it. I can find 7 different diseases on there. No idea if any of those diseases caused the symptoms that caused it to be sent to me. And we do have our sample submission form that has a lot of our site history information that I really like to get from you guys. We also have a QR code as well that will take you to our website. And this next year, we are hoping to be set up to receive online credit card payments in the clinic as well. So, 2019 finally moving into the early 90s. as far as credit cards, but so we're able to get that. Also have a Twitter account. Feel free to check that out. Try to keep people updated as the year goes on, what diseases we're seeing throughout the year, or if people have any questions. I accept pictures, try to answer questions there as well. Just some other resources for the disease management. Crop Watch, great resource, comes out every week during the growing season. Market Journal, you guys are probably aware of that as well. Other one that that might be new to you is the Crop Protection Network. And the Crop Protection Network it's a group of university scientists really from around the country that have gotten together and have tried to solve some of these larger issues. Great resources there on the Crop Protection Network. Also, Dr. Tamara Jackson-Zims, will be a great resource as well. For anyone who didn't know, Lauren Giesler, our former soybean pathologist, he became our department head of plant pathology. And so Lauren is no longer working in soybean diseases. And so this next season, Tamara is going to have her hands full dealing with both corn and beans. But between Tamara and myself, we should be able to help you guys out. With that, any other questions? Yes. Yeah, so it was a question about when planting certified seed or pathogen-free seed in wheat to control black chaff. And the answer is no, they don't. Not all companies do test it. And it can be difficult, and it can be really difficult to prove where the infection came from as well, since that the bacteria that causes black chaff it's fairly common just in the soil. And again, so maybe it was seed transmitted, but it easily could have already could have been in your field. So if you are planting certified seed, that's the best that you can do. Just try to make sure that you have as high quality a seed as possible.
So if it's already in the soil, then that field
it depends. And so all depends, it's in the soil. Think about one of your earliest ecology classes, and you learned about the coyotes and the rabbits. And so the rabbit populations go up, and shortly after that, now we have our coyotes that started increasing. The more coyotes there are, they start eating the rabbits, and the rabbit populations drop, coyotes drop. And so we kind of get that wave-like pattern. The same thing happens in the soil with all of our soil microbes. And so the bacteria, while it is a plant pathogen, there's other things in the soil that will gladly eat that, whether they're nematodes, insects, or just other animals that are in the soil. They'll consume that, and so your populations of that bacterium will kind of vary over the course of a year, over the long term, that long-term growth of the field as well. But environment is going to play a big role too. And so if we have really dry conditions, then we don't expect to see near as many bacterial diseases. If it's pretty moist and you have a field that has had issues with black chaff, well, it might come back. So it's a tough one, unfortunately. Yeah.
Kyle, with the bacterial diseases, what's your opinion on the copper hydroxide product? That's not all we've got.
Yeah, so the question was, with our bacterial diseases, some of our copper products, copper hydroxide, are about the only thing that we can do. At least with bacterial leaf streak, we have not shown that with the trials that we've done at UNL, we haven't shown that to be effective.
Has UNL looked at hydrogen peroxide?
Not in a field study. We've done some greenhouse studies on that, and unfortunately, we did had a really big multi-year study with bacterial leaf streak that we started last year, and then we found out last fall that the funding got cut for it. So It's Tamara's lab is trying to answer a lot of those questions, and we've kind of working through the data. But right now, we haven't been able to show that they are really effective. Anecdotally, have definitely heard from people that they work, but there's typically not a control, or not the control treatment that we're looking at as well. Now, bacterial pathogens, a lot of those we tend to think about some of the larger agronomic practices, and so resistant varieties are going to be really important. Anything you can do to improve soil drainage will really help with some of those bacterial diseases as well. Any other questions? Oh, all right. Well, I also have a few toys that I brought out. One of them is we have some Riker mounts, and these are just dried leaves that have different symptoms of different diseases on them. For those of you who can see this one, any idea what it's the soybeans, but any idea what might cause kind of the interveinal lesions like that? Yeah, SDS, I heard. This is actually herbicide injury. This was injury caused by a triazole. So again, the pathogens, they don't read the textbooks. They don't really know what the symptoms are supposed to be like. Just because you see something, that doesn't necessarily mean that's what the problem is. Also have a few some corn samples and some scopes as well for anyone who would like to come up and look at some samples. But one of the things that I saw a lot of this past year was Nigrospora ear rot in corn. It's a fairly minor, again, generally it's a fairly minor pathogen or fairly minor ear rot. We saw a lot of it last year. Why was that? Well, when you think about when we had all that moisture, the ears were still upright. Moisture, the rain comes, just sits in there, soaks those cobs, and now we're able to get a lot of those ear rots. You look at it from the outside, everything looks fine with it, but and again, you'll probably have to come up here to see these more closely. But as soon as you then start to look at the individual kernels, just get all sorts of these kind of black little streaks at the base. We saw a lot more of that last year. Have some Frogeye samples that you guys can look at. These samples are actively sporulating as well. And so I can make a tape mount. You can look at the spores, the Frogeye spores if you would actually like to. And I have some bacterial leaf streak, which should stream for us if anybody would want to see that, as well as just a few different stalk rots that we've gotten. So you guys are welcome to come up and look at stuff. Discuss any issue, or if you guys have any specific issues in your field, I'm happy to kind of work through those with you too.
Let's give Kyle a hand for his presentation.